Now to introduce our speaker, Alex Zumberge. Alex is the Director of Analytical Services at Geomark Research and manages oil technical service projects, as well as the new GRI Plus Rock Lab. He has extensive lab experience in oil and rock extract analysis with emphasis on correlation tools like bulk organic properties, lipid biomarkers, and carbon isotopes. Alex recently finished his PhD in organic geochemistry at the University of California, where he specializes in sterain-hopane relationships from the Precambrian to present. And I'd also like to mention that another one of our great RMAG members, John Curtis, with Geomark, is also co-authored on this abstract. And I hope John's here today too. So hello, John from RMAG. And with that, take it away, Alex.
Okay, thank you. Let me share my screen. Okay, can everyone hear me and see my screen?
Yes, Alex, I can hear you and see your screen and see your pointer.
Okay.
Thank you.
Okay, so on behalf of Geomark and my co-authors, John Curtis and John Zumberge, first off, thanks to John Ladd for the invitation to give this presentation. And also many thanks to the RMAG organizing committee, including Debbie, Kathy, and Dan for helping me get set up and ready for today's talk. So today I'll be discussing geochemical correlation of late Mississippian sourced crude oils from the Western USA. We'll mainly focus on three areas: the North Dakota Tyler Heath oils from the Williston Basin, a few distinct families from central Montana, Heath evaporites and carbonates. And lastly, we'll do sort of a mini case study down here in southeastern Colorado near the Las Animas Arch. Okay, so before we dive into the data, I want to do, I want to introduce and maybe do a review of what exactly we're measuring from these crude oils, and I won't ask anyone or quiz anyone on what, to have a guess of what they think this might be. It's not a candy or anything like that. It's actually an artist rendition of a eukaryotic cell. So remember way back from grade school, we know that this is a eukaryotic cell since it has a nucleus and some organized, compartmentalized internal organelles. Whereas a prokaryotic cell, like that from bacteria, for example, don't have these components. So we'll focus in on the membrane, the cell membrane up here. There's a lot going on. It's a complex system, but generally speaking, the cell membrane separates the interior of the cell from its outside environment. It protects the cell from its surroundings, and it's a way of controlling movement of substances in and out of the cell. So let's zoom in a little bit more to the cell membrane to see the composition. So it's made up of what we call a phospholipid bilayer. So we have these hydrophilic or water-loving ends on the internal and outside portions of the membrane, with more hydrophobic or water-fearing tail ends in the middle here. And you can see dispersed between these phospholipid compounds are an important class of organic compounds, steroids. So here, for example, is cholesterol. I'm sure we have all heard of cholesterol. This is what the compound looks if you were to build it with what's called the ball and stick view. We can rotate this in 3D space, and you see how it's a nice planar structure. It would fit nicely into the cell membrane in between these phospholipids here. Steroids in general have a few different roles within the cell membrane. Mainly, they regulate cell fluidity and permeability. They can act as structural support, and they can be used even as a defense mechanism. And I'll dive into that a little bit later in the talk. So here's cholesterol, now visualized a bit easier as a line drawing. So each of these apexes or points in the line drawing represents a single carbon atom. And if we add all these up in cholesterol, we have 27 carbons. So if we think about the diversity of algae and bacteria throughout Earth's ocean at any given point in geologic time, you can imagine that we get an ever-changing and ever-evolving and adapting mix of organisms that are each producing characteristic biolipids that help them live in their environment. Whether that's a distal marine setting or a lacustrine setting or a SABCA or all the different types of environments you can imagine. The point is certain organisms thrive in certain depositional environments, and after burial and deposition of these critters, once they die, their chemical makeup gets buried in the rock, and eventually a petroleum system may develop, and upon carriage and maturation and generation and expulsion of crude oil, these biomarkers can be measured in the lab and give us a glimpse into the conditions of these ancient paleo environments. So unlike other very source characteristic or specific organic compounds like proteins and carbohydrates and DNA, steroids are actually preserved on geologic timescales, since they're mainly composed of these very stable hydrogen-carbon bonds. You can see the other compounds have a lot of potential reaction sites here, so they easily break down during diagenesis, whereas again, these steroids are well preserved and very recalcitrant on geologic timescales. So our example biolipid here of cholesterol would get buried at the water sediment interface. It would continue to get buried and go through diagenesis and transform ever so slightly into cholestane. So cholestane is again a biomarker with 27 carbons, a very common one, and you can think of these guys as molecular fossils in some sense. So generally speaking, bacteria will characteristically produce these BHP compounds within their lipid membranes. Those compounds are buried and preserved in the rock record as hopanes, whereas eukaryotes produce steroids, which, as I mentioned, are buried and preserved in the rock record as steranes. So at any given time, you can measure the relative abundance of steranes to hopanes and get a good idea of the algae to bacteria, from a snapshot in time. And I like this graphic because it reminds us of the importance of food webs and the trophic levels. So if you ever find yourself eating a nice tuna sandwich, just realize that 100 grams of tuna required 10,000 kilograms of primary producers. So likewise, the biomarkers that we're measuring and that we routinely measure in the lab from these rock extracts and produced oils are coming from our foundational level organisms, phytoplankton, primary producers, algae, bacteria. So here's an RNA tree of life modified from the famous Carl Woese. This just depicts different domains of life will again characteristically produce different biomarkers, and when all of these are added to our geochemical toolkit, you can think of it as, we can more accurately describe ancient depositional environments. This figure is amusing too, because it's always humbling to see that us as humans up in the animal branch up here, we're only a few blocks away from the slime molds, which maybe isn't too surprising these days. But anyways, you get a general biological input into your proto-petroleum system, you can think of it as, and then those different inputs, right, will leave behind characteristic and unique geochemical compounds for us to then measure in the produced oils or rock extracts. And with all these compounds, again, we can reconstruct certain things like the depositional environment and even the thermal maturity of the organic matter we're looking at. So hopefully that was a useful review. It's always nice to just always put it back into context and see what exactly we're measuring when we talk about biomarkers. So here's, let's get into the meat of the talk now. Here's a paleogeographic map of the late Mississippian, so approximately 325 million years ago. So I've circled this area in red here, and this particular area would be an ideal setting for the deposition of potentially exceptional source rocks. We have a shallow interior seaway that could harbor high productivity from upwelling zones where nutrient-rich waters, right, induce algal blooms of primary productivity. There's also the potential for exceptionally good preservation of the organic matter via areas of photic zone euxenia, or PZE. And this is where we get free hydrogen sulfide in the photic zone of the water column. I'll touch on this at the end and go into more detail then, but piggybacking off of the intro slides, right, the concept's the same. There's certain bacteria that thrive in certain environments, and when the conditions are right, specifically during photic zone euxenia, those bacteria create these certain organic compounds that we can then measure in the lab, and then again lead us to say something about that environment. So for this study, as I mentioned at the start, we'll mainly focus on central Montana. We'll be looking at oils from central Montana, western North Dakota in the Williston Basin, and the Los Animas Arch area of southeastern Colorado. You'll see some other data sets plotted here and there, but these three will be the main areas of focus. So I know it's taboo to show a complex table like this, so I'm just gonna, let's see, gray some of it out and let's highlight a few different types of phytoplankton here, right, primary producers. And this just shows you that different phytoplankton classes inherently make steroids with different compositions. And mainly that's in the total number of carbons in the structure. So as I said, cholesterol has 27 carbons, but we can get steroids all the way up to 30 total carbons. Red algae, for example, these bangiophytes here, predominantly makes steroids with 27 carbons. From some of the more common green algae, the chlorophytes and chrysophytes, those guys are making more steroids with 29 carbons. But there's a special class of green algae down here called the prasiniophytes that only produce C28 steroids. So, right, steroids with 28 total carbons. Hopefully you'll see why I'm bringing this up now, because later in the talk, we'll revisit these prasiniophytes and see how they could be a telltale sign of some of our late Mississippian sourced oils. So if we look at the relative abundance of some of these sterines through time, you can see that the ratio of sterines with 28 total carbons versus those sterines with 29 total carbons is generally increasing as we go from deep time to the modern. On a high level, sort of big picture scale, this has to do with the evolving, the ever-changing and evolving makeup of the dominant primary producers throughout Earth history at any given time. So early in the Paleozoic, green algae were the dominant guys in the ocean, and as I said, most of the green algae will predominantly produce steroids with 29 carbons. So these values, this ratio of 28 to 29 sterines is low in the early Paleozoic. As we move forward through time and different organisms start to become more competitive and abundant in Earth's oceans, for instance, copelithorids, dinoflagellates, and diatoms, this ratio increases because these organisms will characteristically make or predominantly make steroids in their cell membranes that have 28 carbons. So right through time, this is increasing as the dominant or prevailing phytoplankton class in the ocean at a given time changes. So, this means, right, you can almost take an oil at, from any basin in the world, do a simple quick measurement of this C28 to C29 sterine ratio, and with some degree of confidence get a good idea of how old your oil is. So for example, Devonian Woodford oils in the Paleozoic here, these guys are, if you look at their sterine distribution, they're dominated by C29 sterines, so this ratio is low. If you then go conversely to, like the Miocene Monterey Formation, much younger, diatoms were a major source input there, so this ratio of C28 to C29 sterine is much higher. So that's the sort of the concept we'll be building off of in this talk. So let's go back to our map now, showing the areas of interest of these late Mississippian sourced oils. So from Geomark's Williston Basin oil study, we see this unique cluster of oils shown here in this blue, what we call family 5. These are proposed to be sourced from upper Mississippian Tyler/Heath source rocks. And we can see, right, they're very geochemically distinct from other Williston Basin oil families. This type of figure here is just called a dendrogram. So it's built from taking all of your different oil samples in this case, measuring the geochemical fingerprint of each, and then statistically grouping them into families based on how similar they are or how different they are. And so that's how we can group, again, based on the source or the maturity or what have you, into different families. So again, we'll focus on these family 5 upper Mississippian oils here, very distinct from the lower Mississippian carbonate-rich source rocks, distinct from the Ordovician source rock that have that very unique G. prisca signature, and then even distinct from some of the other Devonian source rocks in the area. We'll shift over now to central Montana. Here I'll highlight just 3 oil families, the pink family 2 from these Heath carbonates, and then family 3 and 4, which are these Heath evaporites. And one of the main drivers that started some of this work was the question that we have these, central Montana Heath oils and we have North Dakota Heath oils. Are they geochemically similar or different? So keep that in mind as we look through the results. Lastly, we'll go down here to southeastern Colorado, look into a study that Geomark did with the Los Animas Arch area oils. So here I highlight family 1, which we sort of subdivided into 1A and 1B. Again, right, these are classed this way because they're geochemically distinct. They have a different fingerprint, different characteristics compared to some of these other oils, which group into different families in this area. So our family 1 are the ones with, again, the high C28 sterions from marine shale source rocks. All right, so we can now start cross-plotting some data. So here's just common pristane phytane biomarker ratio, easy to obtain from just a gas chromatograph of an oil versus carbon isotopic signature. So what we see are sort of 3 major groupings, right? We get our late Mississippian Heath source oils from North Dakota here in these blue dots. We first see immediately that they're pretty distinct and different from these squares over here, the central Montana Heath carbonates and evaporites. So already we're getting an idea to answer that question that I proposed in the last slide, that indeed, it seems that at least from this cross plot, and of course we'll go into many more, that the central Montana Heath oils are different from the North Dakota Williston Basin Heath oils. So, and then, as I said, the other ones in the data set, Fort Worth, Permian Basin, Barnett, these guys are plotting down here in their own group. All right, so now we can plot some more data and compare the relative amount of algae to bacteria, right? We can look at the sterain versus hopane ratio and plot that against this common upwelling biomarker ratio we use. And there's a trend here, if you see it, as you'd expect, when you get increased upwelling, you get more algae relative to bacteria, since those eukaryotic algae primary producers will thrive in areas where you have deep, dense, nutrient-rich water upwelling to near the surface conditions. So this shows the trend here, increased upwelling. We did a collaboration with Dr. Chris Scotese. He's a paleocartographer, and he built this. This is a model of upwelling during the Mississippian. So you can zoom in a little bit here. The takeaway is areas where you have the larger blue dots infer more intense upwelling, and therefore higher productivity, and therefore the potential for some really nice source rocks. So some of our basins are highlighted here, which indeed are very nice source rocks. So this is a cross plot from some extended Hopane values from bacteria or prokaryotes versus gamacerane. Gamacerane is a common biomarker used to indicate water column stratification. So, right, unsurprisingly here, our oils from our evaporitic environments, from these central Montana heath evaporites, are plotting away from these more marine shale type plays that aren't as stratified as an evaporitic system would be. So of all the crossplots I've shown so far, this is the one that really stood out the most to us. This is a plot of the C28 to C29 sterane. So that's the plot I showed through time. And this is what we call the regular steranes versus their aromatic equivalent, just a more thermally mature or more stable equivalent compound. And what we see is these anomalously high C28 to C29 sterane ratios for oils of this age, right? Lower Paleozoic oils. So we're getting values 1.5, maybe up even to 2.5. And again, this is coming from predominantly our North Dakota Tyler Heath oils, and then our Los Animas Arch oils. These two areas have been consistently grouping together in some of the other cross plots, whereas our central Montana data set have always been a bit separate. So, right, I'll put this back into context in the next slide. But remember our friends, the Prasiniophyte green algae. So it turns out that these guys thrive in oxygen depleted environments that might have been unfavorable for other planktonic primary producers. So remember the Prasiniophytes, they're unique in that they only make steroids with 28 carbons. So perhaps seeing such a dramatic spike in this C28 to C29 ratio, such high values, could give us a clue that these guys simply outcompeted these other eukaryotic algae in this perhaps oxygen-starved paleo environment. So here again is our graph, right, of the C28 to C29 sterane ratio through time. If we plot our late Mississippian sourced oils from North Dakota and Los Animas Arch, you see they're way off the charts. Here's 1.0 right here. We're getting values 1.5 to 2.0 to 2.5. So again, very surprising to see that. Very strange for an oil of this age. You'd expect much more lower values. All right, so now I kind of want to shift the focus a little bit and do, as I said at the start, a little case study, you can think of it as, involving 80 or so crude oils that we collected from wells in the southeastern Denver Basin and the Los Animas Arch areas. We figured this would be a bit more applicable to this RMAG audience. So on this 80 plus sample oil set, we did our statistical analyses. We measured the geochemical fingerprint in the lab and then plugged them into the statistics to get different groupings and different families, same concept, again, based on their genetic specific biomarkers, as well as their carbon isotope compositions. And a quick aside from this before we dive into the Los Animas Arch data set, we've been asked a few times over the years if we get any Woodford-like oils around the Los Animas Arch of southeast Colorado. The short answer is no. If we really zoom out and look across central U.S., we can see a series of oils generated from various Woodford source facies, shown here, families 1, 2, 3, 4. So we see this spanning across Oklahoma, into Kansas, parts of Nebraska, and all the way up into Wyoming. But as you see, and as I'll show in the next slides, doesn't seem to be any Woodford-like oils around the Los Animas Arch, at least that we found. So this trend sort of seems to bypass southeastern Colorado. So here again is our cluster analysis of the 80+ oils in Colorado that we looked at in the Los Animas Arch. Again, grouped into the 3 major families with some subfamilies. So I don't want to dive too much into the details of the statistical model, but the concept is that, right, we're looking at measuring different geochemical parameters, or what the model would call loadings, and that's what gives us the variation in the data set, and the different pulls apart different oils by similarities or differences into different families. So you can see from the loadings plot here, if you have an oil with a lot of percent C27 stearins, like these family 3A and B, they're going to plot in this region in PCA space, we call it, principal component analysis. Whereas if you can't really see it, but there's a pink dot here that says percent C28. So oils that have high C28 stearins are going to plot in this region in PCA space. So again, that's our family 1, 1A and 1B from the late Mississippian. A ternary diagram of C27, C28, and C29 stearins will tell us the same story essentially. It shows the different groupings of our different families and where they plot. Again, we put the Devonian Woodford oils relative to these Los Animas Arch oils here to show, right, they're very distinct. There's not very strong evidence of any Woodford sourced in this area. So if you think of these sort of clusters here as end members and you get some dots in between, that could be potential mixing where you're not dominated by one or the other, but a mix of both. Okay, so I pulled a few oils from this study, again, just to highlight the geochemical differences and the makeup between, in this example, a Mississippian sourced and a Middle Pennsylvanian sourced oil. So so you don't have to squint. I've enlarged these, but I'll hark on it again. Unusually high C28 stearins from this late Mississippian, in this instance, Warsaw Formation. Here's a strat column. And that's versus what what we consider a more typical C28, C29 distribution here from this Middle Pennsylvanian Cherokee group right here. All right, so when all the data is acquired and everything goes into the statistical model and it's built out and developed and families are established, we can then plot, right, geographically, just on lat long, where these wells actually are and where the oils were collected. And more times than not, reassuringly, we get a nice, nice divisions and nice groupings of our families in a geographic sense. So along the Los Animas Arch, starting with family 1, we get some good clustering here on the eastern flank. There's potentially some mixed candidates here that are on the western side. Family 3, A and B, seem to be pretty geographically well defined from the western flank, family A, 3A, versus those in family 3B. And then up here, these distinct little family 2, again, might be some mixing with family 1 or 3. Okay, I think what time is it? Yeah, okay, I still should be good on time. So before I can conclude, I wanted to talk a bit more about PZE, source rock depositional environments. So this is a cartoon showing euxenia in the photic zone of the water column and how we might get to these conditions or what it takes to get to this. So it all starts with sulfate reducing bacteria. These guys will fix sulfate and produce hydrogen sulfide. That hydrogen sulfide is then used as a food stock for green sulfur bacteria. And for them to use hydrogen sulfide and use it as energy, light is required. in this reaction. So for these, for these guys to be abundant and thrive in here, there's a couple things and conditions that have to be met. So just like I went over with our eukaryotes, some of the algae, the concept is the same here. These green sulfur bacteria produce this unique class of organic compounds called carotenoids. And just like steroids, these carotenoids are recalcitrant on geologic timescales, and we can measure them in the lab with our mass spec instruments. Again, as I mentioned, right, by sort of the laws of this reaction here, that kickstart this equation, this reaction, you need hydrogen sulfide, and they're strictly anoxic, and they need light as well. So without those conditions, these guys wouldn't be here. And so when all conditions are met, that's when we get sort of the extensive photic zone euxenia. So low oxygen, high sulfur, and photic zone obviously implies shallower up in the water column. So again, these can be measured from a rock extract or an oil. And if we see these guys in these compounds, either as whole chunks, so like a C40 compound, the carotenoid class I mentioned, or as they break down, they break down into these smaller chunks here called aryl isoprenoids. This is another sign of PZE. Again, if these are abundant, we can infer that the conditions were met for these green sulfur bacteria to thrive, leave behind their chemical fingerprint, and for us to measure and then make an inference about that depositional environment. So a relevant question, I think, in this instance, and this, what ties to this data set would be, our PZE conditions causing the exceptionally high C28, C29 stearin ratios we see. So here's this cross plot again of algae to bacteria, stearin to hopanes, versus our upwelling signature. We have our, again, in the boxes, Central Montana, heath carbonates and evaporites. Down here we have the North Dakota heath late Mississippian oils, and then the Los Animas Arch in the diamond figures here. So this is our sort of our first separation of the North Dakota heath oils and the Los Animas Arch, and all the other cross plots, right? They were grouping together, and the Montana sample set was distinct. So now putting this in context of, right, we know these guys down here, the Los Animas Arch and the North Dakota, both of those have the anomalously high C28 to C29 stearin ratio. But when we look at for the presence or absence of those carotenoids and aryl isoprenoids, again, indicative of photic zone euxenia, we see that Los Animas Arch oils are absent of those, whereas the North Dakota heath in the Wilson Basin, lower Mississippian, late Mississippian, have abundant PZE biomarkers. And then likewise, it's hit or miss for the Central Montana data set too. So all this to say is that PZE has been recorded across Earth history, and it's a more common than you would perhaps think. It does lead to really good organic matter preservation after burial, but it doesn't seem to be the cause of this anomalously high C28 to C29 stearin ratio. So again, I think the theory that a certain algal class, like perhaps these prosceniophytes, it still holds true that maybe in this scenario, these guys just could have outcompeted other eukaryotes or other algae in these oxygen depleted environments, but not necessarily ones with euxenic conditions. So to wrap up and conclude, got a couple minutes left. Compositional differences, right, observed in our late Mississippian sourced oils across North America. We were able to measure these geochemical fingerprints, group them into certain families based on their similarities or differences, and then see how they compare in a large scale setting. So, right, differences in the depositional environments, the dominant primary producers at the time, whether there was water column stratification or upwelling, or some extreme redox regime like PZE. We had exceptionally high C28 to C29 stearin ratios in the North Dakota heath and Colorado Los Animas Arch oil sets. Again, proposed to be sourced from these prosceniophyte green algae that thrive in these oxygen stressed environments, maybe just outcompeted any other phytoplankton in those environments. And again, they're only producing sterols with 28 carbons, which makes them unique. Largest values known at this time in Earth history for that late Paleozoic timeline. So that's when I plotted those values against the general trend. They're off the charts. For an oil of that age, we would not expect such high values. Lastly, right, the question I asked at the start, are heat source oils from the Williston Basin, are they similar or different from those of central Montana? And they are strikingly different. So there's no evaporitic signatures in the Williston Basin. There's the low stearine-hopane ratio versus high, and then the differences in the extremely high C28 to C29 stearine ratio between the two locations. So with that, that concludes my talk, and thanks for your time, and let me know if there are any questions.
Yeah, thank you very much. I'll kick us off with a quick question. I'm kind of getting old.
You're a little choppy, but okay, I think I can hear you now. Go ahead.
Okay, let's try it. I'm such a visual person, and I'm not a geochemist by enough nature here. You know, going back to the initial paleo environment map that you showed, and also the map with trying to predict upwelling. I mean, have you taken the time to either cross-reference those maps or update the paleo map with SCOTC? Either validate or change the details of some of these intercratonic basins?
Yeah, so this was a GeoMark effort before my time, but it's kind of exactly as you're describing, different slices throughout Earth history, modeling these different basins, and then plugging in what we know from the biomarker analysis, and seeing if it makes sense. Is that more or less the goal of it was? So yeah, there was a big effort.
This is paleoclimate.
Yeah, this is paleoclimate.
Based on different mountains and ocean.
Yeah, and I got my co-author, John Zumberge, here next to me. But yes, we have, that was part of that effort to sort of, go through different major periods of Earth history, do the paleocartography, the mapping, and based on the biomarker analysis, or complementary to the biomarker analysis, see how this makes sense. And again, if you're near the equator, that's just on any spherical planet with water, that's where that rotates. That's where you're gonna get your high areas of upwelling. So these basins were in the right place at the right time with the right conditions to give you such good source rocks.
Okay, yeah, thank you, Alex. Question coming in for you. It says, excellent analysis and report. Thank you very much. What is the success in mapping and explaining the microenvironments from subsurface cores and logs, that is?
Mapping and explaining the microenvironments. So, yeah, these are, keep in mind that these are all from oils, and oils are they're not as precise. They're more of a mixture or a overarching signal of what you're looking at. We haven't incorporated any log analysis to any of these studies. We're now getting to be a little bit more petrophysical, so could potentially incorporate that later on. But this study and this story was built around oil analysis. Nothing with logs or log analysis or anything like that.
Okay. Can hopanes discriminate between bacteria versus archaea?
So yeah, archaea will have their own. Archaea is kind of the one, not an underdog, but they're kind of the forgotten clade, I think. really, really strange critters that live in, in Yellowstone hot springs and whatnot. Very extreme, unique environments. Yeah, they're extremophiles is a nickname for some of them. But generally, yes. Again, there's a little bit of caveats, but general rule, as I said, bacteria are producing hopanes, eukaryotes are producing steranes, archaea then are sort of their own little unique class of compounds. But definitely, in my opinion, understudied and definitely not on the same, don't have the same understanding as eukaryotes and bacteria.
Okay. Question from Holly. I would like to know a few more details about how the oils are sorted into families. Are many more parameters used than the few you talked about today?
Yeah, so let me go back to that loadings slide. So in the statistical approach, for this particular study, the Los Angeles Arch, each of these points here in this loading figure is a different geochemical parameter or fingerprint or ratio of two compounds. So yeah, the short answer is yes, we're using many different either biomarker ratios or carbon isotope values or many different parts of the fingerprint you can think of. The data set is more compelling or more convincing with the more even spread you have of these loadings. Again, because when you actually put in the oil, the results from the oil or rock extract, right, it's going to have a certain fingerprint and it's going to get pulled to one region or the other. So the better spread you have of these loadings, which this is a pretty good spread here, the easier of a time you have to show those distinct differences. And I didn't mention on the dendrogram, but there's no y-axis. This is not a strat column. This is just all of the samples looked at. On top here is the correlation coefficient. So if I was really pessimistic, I'd shift this all the way to the right, and I'd say there's one oil family made up of 80 oils. Or I can shift it all the way to the left and say there's 80 oil families made up of 80 oils. So you kind of play with this correlation coefficient until it naturally, you see natural breaks in the data set into these family groupings. So yes, the more parameters, the better. Again, that's helps us visually see how the data gets pulled apart and separated into families.
Yeah, thank you, Alex. Another one. Can you go over again the significance of the MZ133 aryl isoprenoids relative to PZE?
Yeah, so the aryl isoprenoids are a byproduct of the carotenoid compound class that I mentioned. So I don't have a carotenoid drawn out here, but you can imagine, for instance, isorenirotane is one type of carotenoid. It has 40 carbons. It's essentially, it looks like this if you double it and then add it to this chain. So it's a really long molecule. Hard to stay, it's recalcitrant, but it's hard to stay intact like that when it's that size. So when those carotenoids break down, they break down in a characteristic way, in a segmented, fragmented way, where we can measure the aryl isoprenoids, the breakdown products of those carotenoids, and you can see these peaks here on the 133. The 133 is just the mass and the mass spec, the fragment ion of what we're looking at. But the point is, they're breakdown products of carotenoids. You're not going to have, you need to have the carotenoids first. You need to have these conditions first with critters making carotenoids to then get aryl isoprenoids. So again, another clue of photic zone euxenia.
Okay, thank you very much. Another one, Alex and John, thanks for a great presentation. There has been a second Tyler, quote unquote, source rock identified in the northwest part of the Williston Basin in McKenzie County. Has anyone sent you one of these oils to compare with the southern Williston Basin Tyler oils?
Don't know off the top of my head, but
Maybe.
that, yeah, that, Rich, we could follow up on that. And again, it's really easy to that's the beauty of these models. You sort of build this story from the start, and then any new samples you just measure and plug in and see how they relate to these established families. But I don't know off the top of my head, yeah, if the different counties in here, but for sure we can follow up with that.
Sounds like Rich has got something up his sleeve there. Another question, does thermal maturity, burial, or temperature have any effect on the ratios within each family or environment?
Yeah, so that's another good question. So apart from, I mentioned here, the statistics are based on genetic specific biomarkers, but thermal maturity for sure does have a control on these biomarkers and the things we measure and what make it into the model. So from just like here's our geochemical summary sheet, the ratios of these biomarkers will change with maturity, and that would in fact change like those values that we're plugging into the model. So yes, it's both genetic based and maturity influenced.
We use ones that are more genetic.
Yeah. The genetic ones are a little bit more are utilized more, but we definitely keep thermal maturity in the back of our mind, because you can imagine same source making two different oils, one's more mature than the other. That would cause them to be grouped into a different families. So.
Okay, well, that's all the questions I have on our end. I'd like to thank you again, Alex, for being here today and for presenting your research. And thanks for everybody who's on the call today for showing up. I hope that today treats you well and that you're not just glued to the TV and to the internet results coming out. Hopefully you can get outside and do something fun.
And don't be a slime mold, huh?
Yeah. So we'll see everybody next month. Please continue to watch the website, and thank you for being here.
Thanks everyone again. Appreciate it.